Hello students, I am Rashmita Maldar, Assistant Professor in Botany from the MP College of Arts and Science, Miramar, Panji. This program is for BSc First Year Botany, Semester 1. Course code is BOG 101. Course title is Environmental Biotechnology. The title of the unit is Role of Immobilized Cell Enzymes in the Treatment of Toxic Compounds. Module name is Biosensors for Air Pollution Abatement and Odor Control. So the outline for this module is definition of biosensors, components of biosensors, working principle of biosensors, significance of biosensors, types of biosensors, organism involved in biosensing process and applications of biosensors. So at the end of this module, students will be able to learn they will be able to define what is biosensors, list the component of biosensors, understand the working principle of biosensors, learn the significance of biosensors, explain the types of biosensors, and discuss the organism involved in biosensing process and cites the application of biosensors. Coming to the introduction, the definition of biosensors is biosensors can be defined as self-sufficient integrated devices that have capacity to provide specific qualitative or semi-quantitative analytical information. These are analytical devices that consist of combination of biological detecting elements like a sensor system and a transducer. Coming to the components of biosensor, biosensors have three components. First is sensor or detector, second is transducer, and third one is electrical circuit. So sensor is a biological component or biochemical receptor. It interprets with the analyte and signal the change in its composition as electrical signal. Transducer amplifies the biochemical signal received from the detector and alters the resulting signal into electrical signal and display into the electrical screen. So it consists of a signal conditioning unit, a processor or a microcontroller and a display units. Coming to the principle and working principle of biosensor, all the biological material including enzyme, antibody, nucleic acid, hormone, organelle or whole cell can be used as a sensor or a detector in a device, and deactivated enzyme is placed in the proximity to the transducer. So the tested analyte links to the specific enzyme bioreceptor and induce a change in the biochemical property of enzyme. The change is in turn gives an electronic response through an electroenzymatic process. So the electroenzymatic process is the chemical process of converting the enzymes into corresponding electrical signal with the aid of a transducer. So the signal is an output of a transducer, which is a direct representation of the biological material which is being measured. So here the electrical signal is usually converted into physical display for its proper analysis and representation. So the principle is depicted in the stepwise manner. So the union of biological sensitive element and a transducer is responsible to convert the biological material into the corresponding electrical response in the form of a signal. So the output of the transducer will be either current or voltage, which is relying on the type of enzyme. So if the output is current, then it needs to be converted to equivalent voltage. Output voltage signal is generally very low in amplitude and is superimposed on the high frequency noise signal. So signaling processing unit or signal conditioning unit is accountable for performing the process of amplifying and filtering the signal. So output of the signal processing unit is termed as analog signal. Output is equivalent to the biological quantity being measured. Here the analogical signal can be exhibited directly on the LCD display, but usually this analog signal is passed to a microcontroller where the analog signal is converted into the digital signal. Coming to the significance of biosensors, biosensors have rapid and continuous measurement rate. Then it has a high specificity rate. They make use of very less reagent, which is required for the calibration. It has fast response time and the ability to measure non-polar molecules can be estimated by other conventional devices. Coming to the types of biosensor, we have five different types of biosensor. First is electrochemical biosensor. Second one is calorimetric or thermal detection biosensor. Third is optical biosensors. Fourth is piezoelectric biosensor. Fifth one is resonant biosensor. So, electrochemical biosensor is an analytical device in which a specific biological recognition element, that is the bioreceptor, is integrated within or intimately associated with the electrode that converts the recognition event to a measurable electrical signal for the purpose of detecting a target compound. So, electrochemical biosensor is divided into three types. First one we have is potentiometric biosensor. It uses the ion selective electrode to convert the biological reaction to electronic response. Second one is amperometric biosensor. It is based on the measurement of the current. Third is conductimetry biosensor. It measures the electrical conductance resistance of the solution. Second we have is calorimetric or thermal detection biosensor. Calorimetric biosensor measure the change in the temperature of the analyte solution. It interprets in terms of analyte concentration in the solution. Third one, we have optical biosensors. It measures the change induced in the intrinsic optical properties of the biosensor surface. It is the most advanced biosensor involving bioluminescence uses, which makes use of the luciferase enzyme for the detection of bacteria. So this is the diagrammatic representation of the optical biosensor. Then fourth one we have piezoelectric biosensor. It works by measuring the change in frequency which occurs when the antigen binds to the antibody receptor. It is a suitable tool for diagnosis of the infectious diseases. Fourth one we have is resonant biosensor. A transducer like an acoustic wave can be connected through a bioelement. The analyte molecule is connected towards the membrane, then the mass of the membrane alters, and finally the change within the mass subsequently alters the transducer's resonant frequency. We are coming to the organisms involved in biosensing process. First one we have bacteria as biosensors. So the microbes respond quickly to the environmental changes like temperature, pH or the presence of contaminants including heavy metal. So these characteristics make microorganisms a good candidates as a pollution indicators. So the biosensors make use of various bacteria. Example is Vibrio fischeri. This is a bioluminescent bacteria which makes use in pollution control in the biosensing process. Second one, we have small mammals as biosensors. Small mammals are frequently used to monitor environmental contamination with the heavy metal. These animals have been used mainly because they are found in the intermediate position of the tropic. So small mammals allows to make inferences about the bioavailability and bioaccumulation of the heavy metal. So example here we have is Apodamus sylvaticus, which is a common long-tailed mouse which is used in order to monitor the heavy metal pollution in polluted site, and it is used in biosensors. Next one we have lichens as biosensors. They are the symbiotic association of fungi and algae and have been widely used in biomonitoring of air pollutions. Lichens are effective organisms as biosensors in order to detect heavy metal in the environment. So we know that they are widely used in biomonitoring of air pollution. They tolerate high concentration of heavy metal by sequestering element extracellularly in order to produce lichen acid complexes. So here we have the example of lichen which is used as a biosensor. It is Xanthoria peritina. Next, we have plant as biosensors. Plant species have the ability to absorb metal through their root system from the soil, water, and overground vegetative organs from the atmosphere. This chemical is in some plants may be transported or may be transformed or they may be stored or accumulated in different cells and plant tissues. So, genotoxicity or DNA damage is an early effect of biomarker at the molecular level that has been used in several plant species in order to detect the pollution in the polluted site. So example is Bacopa moneri, which is a plant which is used in biosensing process. Coming to the applications of biosensors are used in clinical and diagnostic application. It is used in medical diagnostics and clinical biochemistry. It is used in medical self-testing. It is used in medical instrument for cancer and genetic tests. It is also used in agricultural industries to compute the levels of pesticides, herbicides, and heavy metals in the soil and groundwater. It used to forecast the possible occurrence of the soil diseases. The industrial applications are it is used to measure carbohydrates, alcohol, acids, etc. during the quality control process. It is used to check fermentation during the production of beer, yogurt, and soft drinks. Environmental applications of biosensor, they are useful for determining the type and concentration of contaminants present in an environment. They are the useful tools to study the extent of chemical pollution. These are my references for these modules. Thank you very much.